Good afternoon, everyone. Thank you for joining us. Today, we're really thrilled to be offering a materials and methods sort of workshop and educational section, which is titled From Basic Science All the Way to Breast Oncology. And what this really seeks to illuminate is all the incredible technical advances in breast oncology, going down all the way from single cells and drugs to organoids, as well as patient sample analysis. So I think we should just get started. Our first speaker is Dr. Calvin Kuo from Stanford, who's going to be speaking on organoids.
Great, thank you for the kind introduction. I will try to work the AV here. Let's see. Oh, green button is probably advanced. Okay, these are disclosures. And yes, so I'd like to tell you about some of our recent work in organoid technologies. We primarily work in the area of adult human organoids, and these can be now generated from essentially any tissue of the body, and they can be subdivided into a more simple epithelial-only type of organoid, which is more commonly used, and then kind of an emerging technology in terms of epithelium plus stroma, which has many applications for replicating the tumor microenvironment. So I'll touch briefly on both of those. Starting off with the more simple models, epithelial-only. And we've had some very long interests in organoid culture. And this is some of our earliest work, just showing that, for instance, intestinal organoids could be generated and cultured in dishes for hundreds of days. And you can hopefully see that over time, the organoids are elongating into even tubular-like structures, which the intestine tends to do. So now, fast forward, it's now really possible to generate such simple epithelial-only organoids from almost any tissue of the body. This is about half of what our laboratory does, and I'll say this is also possible for mammary tissue. Typically, these epithelial-only organoids can be multiply passaged. They have multilineage differentiation. They have stem cell populations within them. And so over the years, our group has really been trying to advance the field of organoid technology in terms of the culture conditions and growth factor additives. These are really, our efforts have largely focused on the Wnt pathway, which is kind of a secreted morphogen that works through beta-catenin signaling. And either on the left-hand side, using bioengineered Wnt analogs, which are more easily used than Wnts, or our spondins, which are hormones which are not Wnts, but converge onto the Wnt pathway to amplify the canonical beta-catenin signaling. Both of these we have found are very, very useful for organoid growth in general, regardless of tissue type. And these reagents are in various forms distributed throughout the world. So we realized early on that the wild-type organoids would be very, very useful for cancer modeling, in particular, functional validation of oncogene and tumor suppressor mutations. And if you wanted to do this in vitro, conventionally, what one might do is use a two-dimensional cancer cell line that would be cultured in plastic. And as we know, these cell lines can be very, very robust and easy to passage, but they have a lot of pre-existing mutations. And over the decades that they've been used, they may have drifted from their original genotypes. And so if you were to insert a tumor suppressor mutation or oncogene mutation, you're doing so in the context of something that's very, very complex that already has maybe hundreds, if not thousands, of pre-existing alterations. On the flip side, if you used wild-type organoids representing normal tissue, you could build cancer from the ground up and insert those tumor suppressor and oncogene mutations, and then perhaps have a better signal-to-noise ratio in terms of looking at effects, and also to be able to study the initial events of transformation. So we've done this over the years. This is some of our earliest work, and here we took 3 different gastrointestinal tissues as organoids, ranging from colon and stomach and pancreas, and inserted various types of mutations that are common to those various adenocarcinomas. And you can see that there is very strong in vitro transformation in the dish, but furthermore, that you could take those organoids and actually transplant them in vivo and get tumorigenesis, really demonstrating that indeed we have converted normal to cancer in the dish, and we used organoid technology to do this. So fast forwarding, we've done over the years many other types of gene manipulations. We were very, very interested in the ARID1A mutations that are quite prevalent across solid tumor types. And ARID1A is, of course, a component of the Bath chromatin remodeling complex. And really no forward genetic model had been developed in human cells to study the transforming effects of ARID1A mutation. So we undertook this. Yun-Hung Lo, who is now running his own lab at MD Anderson, did sequential CRISPR of p53 to knock it out, and then ARID1A to knock it out within human gastric organoids. And you can see that the ARID1A mutation is leading to increased growth in vitro, increased in vivo tumorigenicity, and a fascinating mucinous metaplasia phenotype that Yun-Hung was able to find was dependent upon ARID1A mutation, dysregulation of the Wnt pathway. But this mucinous differentiation we were able to confirm in tissue microarrays representing hundreds of human gastric cancers, really indicating that when ARID1A is mutated in human gastric cancer, as well as now evidence in other cancers, that there tends to be a mucinous differentiation phenotype. Now, organoids in general, I'll mention, can be useful for small molecule screening as well. And we have been able to, in collaboration with Haiyan Fu's group at Emory, who's really done a wonderful job with this, to multiplex and miniaturize organoid cultures down to 1536 well formats, so very, very tiny formats. And you can see here the bright field of a single well of a 1536 well plate. And these can be plated with high robustness and a very good well-to-well variability or lack thereof, and we can screen compounds. We've done maybe up to 12, 20,000 compound screens. Here we were looking at compounds that were synthetic lethal with the ARID1A mutation. So a recent collaboration has been really wonderful with Christina Curtis, who does a lot of breast oncology work. Here we teamed up to look at tumor genomic evolution in gastric cancer, and in particular to look at the fitness and clonal competition that occurs in the very earliest evolution of cancers, when the primary driver mutations are first being established. And we realized that organoid technology, in which, as I mentioned, you can insert these mutations into wild type, would be a perfect kind of playbox to sandbox to look at these early mutations and their clonal competitions. And so Casper Carlson, who's a joint postdoc between myself and Christina, took our pre-existing gastric organoids that we had already knocked out for p53 in the prior story and monitored these over 2 years in culture. And these are Manhattan plots of the aneuploidy, the SNVs that are present in the organoids. And you can see that at wild type, we're starting off with 2 straight lines indicating that the genome is diploid. But over hundreds of days, you can see troughs developing, representing deletions, peaks representing amplifications. And using omics methods and bioinformatics, you can really infer clonal populations that successive waves that are taking over repeatedly, like the gray clone is succeeded by the red clone, is succeeded by the yellow clone, and going down on that diagram to the blue clone. So there's an incredible a battlefield that's happening that's really invisible unless you look carefully. And furthermore, you can use a lentiviral bar-coded technology to molecularly label each individual cell in the population such that on the left-hand side of these very colorful graphs, you can see many, many colors at early time points. But as you move to the right-hand side, you can see that the red starts to predominate, indicating that a red clone has taken over. And again, using omics type of methods, we can look at the transcriptomes, the single-cell transcriptomes of the quote-unquote winning clone and determine what helps to determine its overall fitness. Now, we've been thinking even more broadly for organoid technology in cancer in that we realize that we can make organoids from many, many different tissue types, and this can be multiplexed across data sets from TCGA and other large-scale genome studies of cancer, in which there are long lists of genes that are amplified, deleted, mutated, methylated, and that we could essentially use the organoids as a system for functional genomics validation of these putative driver oncogenes using any number of the readouts listed on the bottom of this slide that organoids can entail. And so this is a paper that just came out, again, a great collaboration with the Curtis Lab, in which they performed a bioinformatic analysis of several solid tumor histologies in TCGA. And these were picked because we had corresponding organoid types, as I'll mention in a second. But the idea was to mine these for what are called amplification outliers, in which we are looking for the intersection of an increased copy number and simultaneously an increased mRNA expression. And that Venn diagram in the middle is called an amplification outlier. And so here you have six plots describing the outlier landscape for these 6 solid tumor types for which we had corresponding organoids. And so we're going to screen these data sets in their respective organoid type, as illustrated in this slide. So for instance, for PDAC, pancreatic ductal adenocarcinoma, we know this is 95% KRAS driven. So we started off with KRAS mutant pancreatic organoids and then inserted a lentiviral barcoded cDNA library, an ORF library, representing the amplification outlier candidates. Colon cancer, we know, is 85% APC, so we started off with APC mutant colon organoids, and so on and so forth. Great collaboration with Bill Hahn. And so this is an example of screening data in which if a certain ORF cDNA leads to increased growth, its barcode linked in cis will become more and more abundant in the overall population. And so the ratio of day 0 barcode to day 50 is going to be more than 1. And so you can see in this kind of box plot on the top that, for instance, FGF3 has a ratio of maybe 12 or something like that. And so this really indicates that it is growth enriching and was a candidate oncogene hit for this particular screen, which was in esophageal p53 null organoids. And we could put FGF3 back in to these organoids, make them grow bigger in vitro, make tumors grow faster in vivo, treat those tumors even with FGF receptor small molecule inhibitors. And this is very relevant as this is an amplicon in breast cancer as well. So switching gears to the last part of the talk, just epithelial plus stromal organoids. We've been moving towards increased complexity, trying to build in the tumor microenvironment. And we can do this from many, many anatomic sites of the body as tumors, all with their distinct histologies. What we wanted to do is replicate the tumor microenvironment that includes the fibroblasts, the immune cells, the blood vessels, and we've been able to do so. In terms of our technologies using fragment cultures to grow intact tumors as fragments that will contain all of these stromal elements in situ. And so here's 2 examples of organoids, a colon adenocarcinoma, what we call PDO, patient-derived organoid, on the left, non-small cell on the right. And the yellow components are the CAFs, the cancer-associated fibroblasts. The purple are the tumor cells, but we're growing them all as an intact entity. And we're not reconstituting, we're not, say, taking pure tumor cells and adding fibroblasts together. We're growing an intact fragment of tumor and just keeping the CAFs intact within that fragment culture. So where things got really interesting was when we started to look at the immune microenvironment. And we were very pleased to see that this type of fragment culture would preserve the tumor-infiltrating immune populations. And so on the left-hand side, we see TILs, the tumor-infiltrating lymphocytes, tumor cells in purple, but the CD3 positive TILs are in yellow. And you can see the nice membrane staining for CD3. On the right-hand side are TAMs, tumor-associated macrophages, and again, they are just baked in intrinsically into the organoid fragment culture, and this is CD14 positive macrophages. And again, to reiterate, this is not reconstitutive. We're not taking pure tumor cells and adding pure immune cells and mixing them together. We're taking an intact piece of tumor, culturing it as a whole, and just preserving those immune populations in situ. So now, we can start to look at the immune complexity of such organoids that we're generating by single-cell analysis. And you can see that in the organoid, there is a whole diversity of immune cells, not just T cells. I mean, within the T cells, there's many types of T cells. There's helper and cytotoxic, there's very important exhausted T cells, there's Tregs. But in addition to all that, there's B cells, there's NK cells, and there's also macrophages. Typically M2 type. And this is very representative of the diversity that is in the fresh tumors. If we look even deeper at T cells and look at the T cell receptor sequences, we can actually find that the clonotype diversity of those TCRs is really beautifully replicated between the organoid and the fresh tumor. We know we have billions of potential TCRs, and to actually have the same representation of those TCR sequences in organoid versus fresh tumor, we were very pleased to see. And the exhausted T cells segregate to, the enriched TCRs segregate to exhausted T cells. So now with this type of holistic representation of the tumor microenvironment, we can start to add checkpoint inhibitors in vitro and get, for instance, TILs to be activated and to start to kill tumor cells. This is run by J.T. Neal, who's now a group leader at the Broad Institute. And where we're headed towards now is we're moving towards using the system, and we don't have the full answers yet, but efforts are ongoing, in terms of using these systems to be treatment avatars to indeed functionally predict who might respond to tumor checkpoint inhibition. And so here what we're doing is we're getting pre-treatment samples of cancer, we're making organoids. The organoids get anti-PD-1, and then the patients get anti-PD-1, and we're trying to correlate the outcomes. And so this is a Stand Up to Cancer project, and this is ongoing, but I'll say that we're not displeased by what we're seeing. I will also mention that this type of technology can be moved more into mouse systems to take advantage of all the beautiful mouse genetics that exist. And so it's possible, certainly, as we know, to take mouse tumors and to implant them onto syngeneic immunocompetent mice so that the mouse tumor is going to have immune cells infiltrating it. And so when we simply make organoids from those, we can get cultures that have the tumor cells, but with those tumor-infiltrating immune populations, such as the T cells and the macrophages, and they're all just baked in again. So I'll close with one just last bit on trying to extend this even further in terms of immune system modeling, because we really realize that it will be very important to bring in other elements of the tumor immune microenvironment, or that is to say, immune elements that cooperate with the tumor immune microenvironment, such as peripheral lymph nodes, secondary lymphoid organs. And so my good friend and colleague Mark Davis at Stanford has really been advancing a new type of technology to grow lymphoid organoids that can be generated from tonsils or spleens or lymph nodes. And in this type of culture, it's more of an aggregation method, but the immune populations are functional in that you can infect them with viruses such as SARS-CoV-2 or influenza virus. You can quote, immunize them actually with vaccines and then get adaptive T and B cell responses out the back end. And so we really feel that this type of technology has a lot of promise in terms of being able to be combined with tumor microenvironmental models to make even more holistic models of immunity, tumor immunity. And so let me see if I'll stop there, and I'd be happy to take any questions.
Great, thanks. We have multiple microphones. I'll get it started. Thank you so much. This is really, it's so impressive from a pan-cancer point of view how you're able to model all of these different cell types in organoids. I was wondering, many of us in the room study ER-positive breast cancer and how you think about more indolent cell types, and if any of the growth factors and other media conditions that you're experimenting with, if you've been able to identify patterns, and also how you think about growth factors that might be physiologic versus non-physiologic that may enhance cell or organoid growth.
Yep. I will say, in terms of organoid culture, we're essentially growing what we're given. And if it's a high-grade cancer, the organoid is going to grow quickly. Low-grade, more indolent cancers, those are typically, in our experience, more slowly growing. So that does replicate the biology in that respect. If, for instance, the tumor is growing very, very slowly in a person, we may be subject to that as well. But this kind of brings me to the second part of your question in terms of kind of the physiologic niche for the tumor cells. We do try to replicate that. There's a couple different ways. One in which if the tumor is from a certain area, such as the colon, we might try to replicate the growth factors that are present in the crypt area of the colon, which is where the stem cells reside. But on the other hand, as tumors progress and they acquire mutations, there's this concept of what's called niche independency. So they may become progressively independent of the growth factors that their cognate stem cell might have required. And so there may be less of a dependence for that. But at the same time, we do need to find those factors that will sustain the, say, tumor-initiating cells. And I think that's an ongoing challenge, but for many solid tumor types, it is very possible.
Hi. Here.
Oh, great. Yes, center microphone. Thank you.
Yep.
If you could please introduce yourself as
Sure.
well.
Yeah. This is Madhura. I'm a translational scientist from India. I'm a young researcher, just started my lab. So I got the term organoid fragments. Do you mind elaborating a bit on that? What does that mean?
Well, there are a couple different ways to initiate organoids. In one way, you will take the tissue and then you'll disaggregate it completely to single cells. And then you'll let the organoids grow up from that. And we do that method. That's kind of what I mentioned in the first part of the talk. But in the second part, what we like to do is to take intact pieces, if you will. When I say fragment, I mean a chunk, say, of the tumor, and grow that as an intact, what we call fragment. And in doing so, we maintain the architecture of the tumor. Everything's not disaggregated to single cells. We maintain the tumor architecture, the association patterns of the stroma with the tumor cells, and even the spatial relationships of certain immune cell number 1 to tumor cell number 2 is going to be maintained because we're not taking things apart to single cells and then putting things back together. So in that way, it's more of maybe a more accurately representative model of what was in the patient, because we're not just totally unpacking everything to single cells and then reaggregating things back together randomly.
Okay, got it. So is it defined size or number of cells in a fragment, or it's random?
Yes. I would say that several hundred microns.
Okay. Mm-hmm. Okay. Yep. Thank you.
We have a couple questions from online. What is your, I guess this was partially addressed by the last speaker, but what is your ideal amount of starting tissue to get viable organoids, and how long can one keep the tissue in media before processing it?
What was the first part of the question?
The ideal amount of starting tissue.
Oh, sure.
Yeah.
Well, the more the better, but I would say that it depends on the type of organoid that you're trying to make. The simple type of organoids can be made from almost any type of starting material, even as small as FNAs. The larger type, the fragment culture, we like to have enough finite tissue to actually be able to make fragments from. And so there we like to start off with at least a core biopsy, I would say.
Great. And if there are no other questions from the audience, there's one more question from online, which is, sorry. I'm sorry about that. Yes.
Hi there, it's Shom Gold here from Melbourne, Australia. Thanks for a wonderful talk. My lab works a lot with transgenic mouse models of breast cancer, and we've tried to use this fragment system, but we invariably only get outgrowth of epithelial cells in our organoids. And I'm wondering if you have any tips or suggestions of what did you have to do to modify your conditions to get the other cells to persist?
Yeah, we haven't had much difficulty maintaining stromal compartments, but we haven't done breast cancer models. One thing we did remark to ourselves was that, for instance, with calves, with the fibroblasts, sometimes we have the opposite problem. Sometimes they will overgrow and take over things. But then we were thinking to ourselves that we had not done anything in particular to support those calves. And so I would think that if, for instance, if you were looking at calves, and I don't know what you're looking at, but you could use growth factors that would support those populations. So whatever stromal pieces that your components that you're looking at, I would recommend using growth factors that are tailored for those. Be happy to talk to you more offline.
Thank you.
All right.
Thank you very much. Galara Fashid, pathologist, also from Australia, just coincidentally. I love your idea of tumor avatars. So this, automatically leads to whether we can do drug sensitivity for each individual patient. How long does it take to develop an organoid for an individual, and how far away could we be from that kind of individualized drug sensitivity testing?
I think there's two approaches to that. One is when you have a very focused question with maybe just one drug that you're looking at, and in that case, we are not trying to establish a long-term organoid line, but we're using the organoid technology to keep the tumor happy while the drug assay is being run. And in those cases, I would say that you can pull this off within a week or two, pretty easily. Establishing a long-term organoid line that you could then multiplex across hundreds, if not thousands of wells, that takes more time. And so then that, I think, starts to drag you into weeks, plural, to months. And so I think depending on the application, there's a good answer for whatever question it is that you're looking at.
All right. Thank you. I think in the interest of time, we should move on to our next speaker. All of our speakers will be available afterwards, so please come up with your questions. Thank you. Thank you, Dr. Kuo. Our next speaker is Dr. Ashley Laughney from Weill Cornell, who will be discussing single-cell technologies.
Thank you. First, thanks to the organizers for the opportunity to present, and thank you for your attention. My lab really focuses on tackling the genotype to phenotype problem in cancer evolution. What is that? How do we predict function from sequence in complex evolving multicellular systems? And over the years, we have learned that there's a lot of parallels between the metastatic cascade and an injury response, beginning with inflammation that actually emanates from the cancer cells themselves through an ongoing process called chromosomal instability that I'll talk about shortly. And that inflammatory signaling can trigger both immune remodeling of the microenvironment and regenerative processes that are necessary to generate a metastasis in a distant secondary organ site. And so what we see time and time again is that activation of the same pathway can lead to very divergent functional outputs at different stages of disease progression. We focus a lot on studying an immune stimulatory, or I would say immune regulatory protein called STING, where we see early in disease progression, its acute activation can lead to robust anti-tumor immunity, but later we'll demonstrate that its chronic activation later in metastasis can promote disease progression. And there are many other examples of such pleiotropy, for example, with TGF-beta, as well in cancer progression. And our goal is to understand the context in which different functional outputs emerge so that we can best target these pathways in cancer progression. And so we know that emergent function is a product of both tumor intrinsic processes, interactions with the microenvironment, cell of origin, and I'm sure many other complex factors. And today I'm going to tell a single story about how a tumor intrinsic process called chromosomal instability shapes the immune microenvironment to promote immune suppression and metastasis. And so what is chromosomal instability? Copy number alterations have long been associated with the most aggressive metastatic cancers, including breast cancer. They're enriched predominantly in triple negative breast cancers. And on the left here, let's go back. I'm showing sky karyotyping. Sorry, Neil. Of 25 cells, approximately, from the same tumor. This is actually from a glioblastoma, which is highly chromosomally unstable. And what I want you to appreciate here, every chromosome is painted with a different color. And the point is that no two karyotypes are the same within the same tumor. And in a study out of MSK a couple of years ago now, one of the only conserved genetic drivers of metastasis is this fraction of genome altered. So while there is no kind of conserved genomic driver of metastasis at the single gene level, we do see proliferative and many copy number alterations and metastases as compared to their matched primary tumors. And so for many years, it was unknown if these copy number alterations were simply associated with the most aggressive metastatic cancers or were drivers of metastasis themselves. And so almost 5 years ago now, together with the Bakum lab at MSKCC, we basically took otherwise aneuploid cancer models and we dialed up or dialed down the rate at which they missegregated whole chromosomes. models to tease apart this ongoing process of chromosomal instability from the state of having abnormal copy numbers. And what we discovered is that this ongoing process of instability, or SIN, was a potent driver of metastasis. But what was surprising to us was that what we thought this ongoing process of SIN would generate genomic heterogeneity upon which Darwinian selection would act, and that we would identify particular karyotypes that are favorable for metastasis. But what we unexpectedly discovered that through this missegregation process, there was a transcriptional response to cytosolic DNA sensing. So it turns out these lagging chromosomes frequently end up in very fragile, rupture-prone micronuclei. And when those micronuclei rupture, that exposes DNA to the cytosol, which should not be there. That would indicate typically that your cell is infected with a virus. And so the sensor CGAS will detect that cytosolic DNA. It produces a molecule called cGAMP that binds to STING, an obligate homodimer that sits in the ER. And typically we think of this as triggering an inflammatory response. And so the big question was, why is this promoting metastasis? Why is it not triggering anti-tumor immunity through production of type 1 interferon? And so together with the Bakum lab, first we demonstrated that not only does this hold true in fully immune competent models, since most of our initial work was in athymic mouse models with human cancers. So even in the fully immune competent setting, you knock out STING, you suppress metastasis. And remember, this is tumor intrinsic STING. And then when you complement back STING in a dose-dependent manner, you get a dose-dependent increase in metastasis. And not only does this hold true in the fully immune competent setting, but it actually requires interactions with the immune system. So if you look at the SIN or STING-dependent metastatic effect in fully immune competent versus severely immune compromised mice, you'll see that that STING-dependent metastatic phenotype goes away when you lack an immune system, which begs the question, how is SIN shaping the tumor ecosystem? And so what we did was we coupled with the Bakum lab, who are experts in this dialing up and dialing down of rates of instability in chromosomally unstable cancers, and our systems-level analysis of cellular interactions through single-cell transcriptional profiling of the tumor microenvironment. I'm highlighting key contributors here. The main two I'd like to especially highlight are Melissa Hubitz, who is the inventor of the contact tracing method I will present, and June Lee, who is a talented postdoc in the Bakum lab who did a lot of the mouse work and is currently on the job market. So how does SIN shape an immune suppressive microenvironment? Initially, what we did was we took the same isogenic triple-negative model of breast cancer. We dialed up or dialed down rates of instability, and we first just took a static snapshot of the immune microenvironment. And what I want you to appreciate is on the right hand of this axis, we're just showing differential abundance of cell types in a SIN or STING-dependent manner. This is actually SIN-dependent. And we can see in chromosomally unstable tumors, enrichment of these M2 anti-inflammatory macrophages, a large granulocytic MDSC infiltrate, and a lot of dysfunctional T cells. Whereas when we suppress rates of instability or we knock down tumor intrinsic STING, now we see emergence of these more M1 pro-inflammatory macrophages. We also see emergence of some small but mighty subpopulations of dendritic cells, plasmacytoid dendritic cells, CDC1s that are your kind of professional interferon-producing cell types in the microenvironment. And so we see that indeed SIN shapes an immune suppressive microenvironment. But the real question was, how do these cell types interact? And so from single cell data, typically we understand an interaction in the form of there is some donor cell type that secretes a ligand that binds to a complementary receptor on a target cell type. And those ligand receptor pairs are well annotated for both human and mouse. They exist in databases. And I will say most of the initial single cell methods to predict interactions were just that. They were databases. So you would say, is the ligand differentially expressed anywhere in my microenvironment? Is the receptor differentially expressed anywhere in the microenvironment? If the answer is yes to both, then bing, 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 putative interaction. And so you could suspect you're going to get a lot of predicted interactions using this approach. And they're not necessarily all engaged. Just because a ligand and receptor are both present in the same microenvironment doesn't mean they're interacting. And so the next wave of interaction models said, you know what, we're going to prioritize those interactions that show evidence of downstream signaling in the target receptor expressing cell, which was a very good idea. The limitation of these models, though, was that their evidence of downstream signaling was based on very limited prior knowledge. Mainly, most methods use a cytosig database, which essentially was, at the Broad, they took a panel of 6 epithelial cell lines, they added ligand, they sequenced the cells in a dish before and after ligand addition, and they said this is what response to ligand A looks like. What is the problem with that? I mean, one, we know that cell type responses in a dish do not reflect what we see in vivo. And two, we're cell type agnostic. So that means we assume downstream target genes are the same in an NK cell, and a T cell, and a B cell, and a tumor cell. And these are fundamentally different cell types with different responses, different chromatin architecture. And so we had the very simple idea that when there is an interaction, engagement between a given ligand receptor pair, it's going to induce a change in gene expression of the target cell receiving the stimulus. And so by exploiting intrinsic variability that we naturally capture by our single cell readouts, we can actually infer in a data-driven manner which cell types are exhibiting a response to a ligand receptor engagement, and what does that response look like. And so this method we call contact tracing. In its simplest form, it builds upon these ligand receptor databases where for a predicted putative interaction, we'll take the target cell type, and we'll compare those target cells. Let's say it's a macrophage. Let's compare the receptor expressing versus receptor null macrophages. That's going to give us potentially some genes that might be induced by the ligand and genes that are also simply correlated with receptor expression. But by doing that same comparison in the presence or absence of ligand, now we can tease apart those receptor correlated effects from those that may be induced by this given interaction and infer in the in vivo native context, what is this interaction doing to the target cell receiving the stimulus? And that is the basic principle behind contact tracing. I want to show you that we capture known ligand effects. So I'm showing you the predicted fold change in gray in contact tracing. This is for CCR2 expressing macrophage, and I'm highlighting in red and blue known up and down regulated target genes that have been validated, mostly in vivo, actually. Additionally, we've performed very controlled experiments where we sequence, in this case, ApoE-treated macrophages before and after ligand addition. We infer a response, and we compare that to the response we infer from natural variability in the single-cell data itself. And we see that they're highly correlated. We also compared, and this is now in human triple-negative breast cancers, the frequency of our predicted interactions co-localizing in matched spatial transcriptomic data. And what I want you to appreciate is contact tracing predicts a number of unique interactions not detected by other methods, and these frequently co-localize. And we prioritize interactions in a way that best captures their co-localization as compared to database methods alone or methods that rely on prior knowledge, are restricted to prior knowledge of downstream signaling instead of learning that response from the data itself. And so what is the major output of contact tracing? This is called a circus plot. I know it looks complex, but it actually captures all SYN and STING-dependent effects that we capture in this model of triple-negative breast cancer. And so in the outer ring, we encode cell type in which the response is measured or the ligand is produced. And because we actually measure what the response to an interaction looks like, we can organize those interactions in a biologically intuitive way. We do this with something called the diffusion component, but it's computed in the transcriptional response space. We also are able to test whether or not those response states are more or less abundant in a given condition. So it might not be that your response is totally different in a SYN high versus a SYN low tumor, but maybe you have a lot more of a given response state in one condition over another. And that's what's encoded in this color of the third ring. And then on the inside, we have ribbons that connect interacting ligand receptor pairs that measurably shape their target cells in the microenvironment. The width of that ribbon tells you something about the magnitude of the effect, the number of differentially expressed genes, and that target cell receiving the stimulus. And the color of the ligand, the ribbon tells you about the fold change in the ligand abundance. And so those big red ribbons are the ones that are emanating from chromosomally unstable cancer and chromosomally unstable tumors that are really shaping their TME. And so if we zoom in on ligands emanating from the cancer cells themselves, we see that those that shape the TME are actually largely associated with an unfolded protein response to ER stress, as with other pathways associated with SYN, like NF-kappa B signaling and EMT. Conversely, when we suppress rates of instability or knock down tumor intrinsic SYN, now we see that the ligands measurably shaping the TME are associated with that more anti-tumor type 1 interferon response. And indeed, we observe a STING-dependent ER stress response in tumor cells with SYN. Here I'm just showing this for one arm of the unfolded protein response pathway. And additionally, if we take chromosomally unstable tumors and we knock out any one of the 3 arms of the UPR to mediate this response to ER stress, we potently suppress metastasis in these chromosomally unstable tumors, importantly, and as predicted, in a manner dependent on the presence of the immune system. And so in these cancers in which SYN is chronically activating the SABCS STING pathway, we actually see unexpectedly that those type 1 interferon responses are suppressed, and there's an upregulation of an ER stress response. And so our working model is that transient activation of this pathway, which I'm going to show you here in a reductionist fibroblast system. So these are diploid. They have no chromosomal instability. STING is not activated at baseline. If we take those fibroblasts and we stimulate them with the STING agonist, CGAMP, one time, we get a robust interferon response. But when you chronically activate that pathway, which is shown with the red line, you see rapid desensitization or tachyphylaxis, and we lose that interferon responsiveness. Conversely, we see upregulation of this ER stress response. So I really like this analogy of a car alarm. You hear it once, you look. You hear it repeatedly, you're going to tune it out. And that's exactly what the cancer cells do when they're chronically sensing nucleic acids in the cytosol. And so again, our working model, transient activation leads to robust interferon response that's largely restricts tumor growth. But when you chronically activate this pathway, that responsiveness goes down and you see an upregulation of this immune suppressive ER stress response. This manifests in patient samples. So here at the protein level, typically we can classify patients in a chronically activated state. You'll see an abundance of these, it looks like a starry sky. Those are SABCS positive micronuclei that have ruptured and have been sensed by the SABCS STING pathway. And conversely, in tumors that I think would be primed to mount an interferon response, we see that those SABCS positive micronuclei are lacking, and we actually see an upregulation in STING itself. And so I didn't show the data here, but when STING is chronically activated, it actually it mediates its own degradation through autophagy. And you can inhibit that with BAF and prevent autophagy-mediated degradation of self. Which now this opens the possibility of, rather than activating STING in tumors in which chronic sensing of cytosolic DNA is present, perhaps we should actually be suppressing STING. And so, of course, STING has important roles in both the immune system and the host and the cancer cells themselves, which are kind of at odds with one another. So we wanted to test, is this feasible clinically with the drug? And indeed, you can see that inhibition of STING suppresses metastasis, and these are very aggressive metastatic models, in a manner dependent on tumor intrinsic STING. And so with that, I would like to conclude. So how we can exploit intrinsic variability in single-cell data, generate biologically inspired computational methods to identify a context-dependent function, in this case of STING signaling in cancer progression and metastasis. And I'd like to conclude, of course, by thanking the phenomenal lab that I am privileged to work with on a regular basis, our collaborators in the Bakum lab, and our funding sources, and I would be happy to take questions. Thank you.
Great, thank you so much. I was wondering if contact tracing can also be used to infer paracrine signaling that just might happen cancer cell to cancer cell, and how, I guess in your models, are they weighted more to look at the immune cells, or is it much more unbiased and it's just looking for ligand receptor interactions just in all cell types, and whatever is ranked at the top is deemed the most important? And if I guess a second question would be also RTKs and other proteins that may not need a ligand for clustering and for downstream activation, if contact tracing is able to get at those interactions as well.
Yeah. So yes, we can detect autocrine interactions as well as paracrine. There's no bias against that in the data. Can my slides go back up? Is that possible? No. Okay, I also want to emphasize that the code is fully accessible. We've made it open source. There's an interactive web browser, and also you can apply this to your own data and also generate your own interactive web browser for your data. That information is also in the paper if they can't put the hyperlink back up. In terms of discovering ligands, so this is a great extension of the application. So if we can, or think of metabolites, hormones, things that we might not capture transcriptionally in the microenvironment, if we could tell you what receptors are exhibiting a dependent effect, we can make, I think, some pretty good assumptions about what metabolites might be responsible. So I think that could definitely be it's a great, would be a great extension.
Really great discovery tool, as you said, because there are many RTKs. We don't necessarily know all of the ligands, and some of them are orphan ligands as well, or even GPCRs and proteins like that. Yeah. Any other questions from our audience? All right. Thank you so much, Dr. Laughney. Our next speaker is, actually, I think it's Dr. Razavi. Oh, sorry. Is it Sorry, I just want to make sure we're going in order. Yes, our next speaker is Dr. Pedram Razavi from Memorial Sloan Kettering Cancer Center, who will be discussing real-world genomics.
So thanks for the invitation. So switching gears, I will be talking about using patient data and real-world data as a discovery toolkit. These are my disclosures. So I will be talking about 3 different areas. I will talk about our MSK experience with our translational program, and I will highlight a couple of stories. Then after that, I will talk about the large-scale clinical genomic data and how these type of consortiums can be used, and I will highlight the ASR Genie data. And after that, I will talk about AI and machine learning for multimodal data integration. So I think one of the most important part when we started using the clinical genomic testing at MSK in 2013 was the fact that we realized the power of such data, and we started collecting very structured clinical data on all the patients, started putting the data visualization tools and also standardization of the data and clinical data, along with all the other genomic and other modality of the data that were coming out to our database. And then we started thinking about standard pipelines for the analysis to streamline such discoveries. And then one important part of it was the integrated clinical genomic pipeline. As I mentioned, we spent some time thinking about how to decrease the bias in this type of analysis, how to improve the power of such analysis, and what are the modalities of the data that should be included in such analyses. And also we started to include modalities from other than the clinical data that we had. So obviously, the first thing to tackle was endocrine therapy, and this is published data, and I will just highlight the fact that such analysis resulted in identifying multiple novel mechanisms of resistance to endocrine therapy. We highlighted the role of MAPK alterations as acquired and also intrinsic resistance of resistance to endocrine therapy. And more importantly, we uncovered a lot of transcription factors, FOXA1, HERID1A, other members of the SWI/SNF pathway, and also MYC as potential mechanisms of endocrine resistance. There's a significant amount of endocrine lab work now ongoing in collaboration with Tosca Lab at Hopkins to understand this better. I would like to also highlight that there is much work to do, and the important part was that still a lot of the mechanisms were unknown. Then we tackled CDK4/6 resistance, and we identified multiple mechanisms of resistance to CDK4/6 inhibitors. We highlighted the role of HIPPO pathway. This is the work done in collaboration with Sarat Chandlapati. We found that FAT1 alterations result in resistance to CDK4/6 through the HIPPO pathway, CDK6 overexpression, and we also identified multiple other mechanisms of resistance to CDK4/6 inhibitors. More importantly, we started to understand how these type of alterations can be acquired and if this can be predicted. So I would like to highlight one of the stories. This is the work that was done by Anton Safonov, who recently joined faculty at MSK. When he joined the lab, he wanted to look at the interactions between germline and somatic. And he put together a very large cohort of patients with full clinical data, as well as somatic and anonymized germline data, and analyzed the data. So the first thing that he did, obviously looked at the known HRD genes and found that in germline BRCA1 mutation, for example, as reported before, p53 alterations were enriched and p3C mutations were underrepresented. But the size of the cohort allowed us to actually look much deeper into this type of issues, and then probably for the first time, we started to distinguish the signal from BRCA2 and BRCA1, and we found that actually, unlike BRCA1, when RB1 was underrepresented and underenriched in BRCA2, RB1 was significantly enriched in the germline carriers, while P53 was not. And when we looked at the data by the receptor subtype, we saw that most of this actually happening in the HR-positive, HER2-negative subgroup. And this prompted us, this association with RB prompted us to look at the response to CDK4/6 inhibitors. Obviously, it's downstream, results in resistance, and we showed a very significant resistance to CDK4/6 inhibitors on patients who were germline BRCA2 carriers. This is the first-line data, and this is later-line data. And more importantly, we were able actually to use some real-world data. This is the data from FMI and Flatiron, showing that actually germline BRCA2 is associated with significant resistance to first-line CDK4/6 inhibition, and the hazard ratio was very similar to what we identified, confirming the findings that we had from our institution. Then we looked at the mechanism, and then we put together a cohort of paired analyses. These are each dot here shows a paired pre and post, and this means that the alteration was, the border means that the alteration was acquired. And if you look at this, germline BRCA2 mutations had a significantly higher rate of acquired RB1 loss compared to the germline wild type. This was almost 8 times increase, prompted us that there is something going on between the biology of germline BRCA2 and RB. We looked at obviously both genes are in the chromosome 13q and they're in vicinity, and previous work has suggested that as part of the tumorigenesis in the germline BRCA2, large state deletions and loss of RB1 and BRCA2 happens concurrently. We were able to show that actually in our data. This is the loss of heterozygosity looking at BRCA2 and RB1 in the germline BRCA carriers. You see that majority of the cases, they were co-lost together. And interestingly, in the wild type, when there was LOH of that part of the genome, these two genes were lost also together. Around 30% of the patients had RB1 loss of heterozygosity, something that I will deal with a little bit later. Then we thought if the association is because of the germline BRCA2 and RB1 LOH, let's just look at RB1 LOH, and we found that actually RB1 LOH was also associated with significant resistance to CDK4/6 inhibitors and also to overall outcome. We collaborated with Nick Turner and Ben O'Leary, and we are able to analyze Paloma-3 data based on the ctDNA analysis, and their data actually replicated what we found in our single institution. More significantly, the RB1 LOH was associated with very poor OS in the Paloma-3 data. To understand this better, we expanded the paired analyses to look at the effect of LOH. And for this analysis, we separated the LOH to head loss when there is only one functional copy of the RB1 in the pretreatment sample versus other types of LOH when there was gain, genome duplication, so on and so forth of RB. And then we found that almost exclusively RB1 loss of function mutations, acquired ones, happen in the tumors that have head loss with only one functional copy. And this phenomenon is extremely rare in the other types of LOH and also heterozygous tumors, telling that the pretreatment copy number actually can predict the mechanisms of resistance. To my knowledge, this is one of the first examples when we are able to predict the exact mechanism of resistance by looking at the pretreatment somatic data. We think that is probably an expansion of the Knudsen two-hit hypothesis. In the original hypothesis, the patient is born with a defected gene and also loses the other one, the defected allele, and loses the other one through LOH. Here, there is an LOH, and under therapeutic pressure of CDK4/6, cells are more likely to lose that functional copy. So now we're expanding this. We have a large-scale genomic data looking at paired samples and also try to predict the mechanism of resistance and response using large-scale genomics. So in the second part of the talk, I will talk about real-world collaborations, and I will. This is a big effort going both in academia and also in industries, major emphasis on this type of data, and I will highlight some of the data from AACR-GINI. You're familiar with AACR-GINI, but I would like to mention The big part of the AACR Genie was harmonization of the clinical data across multiple institutions. More than 160,000 patients have been accrued to the AACR Genie, multiple cancer types. But they developed this whole system for clinical annotation and standardized it very rigorously. And also genomic data has been harmonized across multiple institutions, allowing us to do a seamless analysis of this data. This is the AACR Breast Genie, and this data is presented for the first time. So the goal of the AACR Genie was to look at the genomic and clinical characteristics of the young women. So the cohort was restricted to patients who were aged less than 55 and age at sequencing at age 65, and also the 3 major centers were included. This is an analysis that is being expanded. I'm just going to show some of the analysis that we've done so far. So what was reassuring was that the landscape in the cohort mimicked what we have known about breast cancer with p53 mutations happening, triple-negative breast cancer, PIK3CA mutations, so on and so forth. So there was nothing, of concern when we looked at the genomic features of the tumor. But when we started looking at the patterns of co-occurrence and mutual exclusivity, we identified some of the associations that have been reported before. But more importantly, we started seeing some relatively new associations, something that has not been reported to this extent before. For example, we found that for the patients who have GATA3 gain of function, this is C-terminus mutations, truncating mutations resulting in stabilization of the gene and increased overexpression of the GATA3. These alterations were associated with, they're mutually exclusive with p53 and PIK3CA and also PTEN, but they were also associated with MDM2, meaning that the cells, instead of losing the p53, they choose to have MDM2 amplification. We are looking into this a little bit more. This association was not seen when we looked at the other loss of function GATA3 mutations. We're expanding this analysis to look at more such associations. Then we looked at the age association. So this is, the cohort was restricted to younger patients. We put the MSK cohort for a comparison, and we performed a trend analysis to see if we can find any trends in the increase or decrease rate of alterations, explaining a more aggressive nature of disease in younger patients. And I'm just showing some of the significant findings here. And in this oncoprint, you see the age categories. And very interestingly, as expected, p53 mutations were associated with younger age. PIK3CA mutations significantly increased by increase in age. And GATA3 in younger patients, CDH1 consistent with lobular breast cancer, more seen in older patients, but started seeing some novel things that, we knew that MAP3K1 and MAP2K4 are a marker of luminal breast cancer, both of them part of the same pathway. We were expecting them to be mutually exclusive, but we were not expecting to see MAP3K1 and MAP2K4 to have differential association with age. For example, here we see that the MAP2K4 is associated more in the younger group of patients than MAP3K1 in older patients. And I'm not showing the outcome data, but also the MAP2K4 seems to be associated with worse outcome. We are looking at the associations, other signatures associated with this as part of the second part of this analysis. And big part of the Genie data was the fact that, site of disease was also selected for all the patients. I'm showing some of the preliminary analysis that we did on the metastatic tropism. We identified some of the known features. For example, ovarian metastases were seen more in lobular breast cancers, bowel the same, and also started to find some associations between alterations and different sites of disease. And we started looking now more importantly into interactions and if this can be predicted from the primary tumor before the patient actually develops metastatic disease. So it's an ongoing analysis. In the last part of the talk, I would like to talk about machine learning and AI and how we can use these features to integrate the multimodal data and real-world data. This is inevitable. I believe in the next few years, we're going to talk much more about this in the meetings and other meetings. So for that, I would like to highlight MSK Mind effort. This is an effort at MSK trying to put together multiple modalities, including clinical, radiographic, pathologic, and H&E and IHC images, as well as genomics, and integrate this data and use deep learning models to predict outcomes. It's the work that is led by Saurabh Shah and Nikki Schultz. And there are multiple publications now in ovarian cancer and lung cancer. I want to show you some of the findings from the breast study. So for breast, we focused on neoadjuvant response and path-CR in this group. And I'm highlighting the team here. What we didn't realize in the beginning was how much of an effort it needs, even in institutions such as MSK, to put such large-scale data and how much data engineering it needs to put this together is something that, if you start doing this type of analysis, the whole anonymization of the data and integration of the data requires significant amount of data engineering. So it's something that was done by the MSK Mind. But then we started to look for to our hypothesis. And one was that can we predict the outcomes using this multimodal data? And more importantly, as a clinician, it was important for me to see if we can predict the outcomes using the clinical data that is already available at the time that we see the patient for the first time, meaning radiographic, pathologic imaging, and clinical data, because now we use very heavily genomic data. And can these be as good as genomic data or maybe even better? So we put together a cohort of 1,200 patients who received neoadjuvant chemotherapy. The patterns of path-CR predicted what we were expecting in this cohort of patients with triple-negative patients and also HER2-positive patients having a higher rate of path-CR. This is the clinical features of the cohort. We are doing more RNA-seq, but the DNA-seq was done. We kept the group who had DNA-seq as our validation set, and then we performed the analysis on the clinical radiographic and pathologic data. So I show some of the unimodal data. These are preliminary data and analysis. We plan to present the full data, hopefully in one of the upcoming conferences. So I think it was very reassuring when we started the analysis that decades of clinical research in breast cancer has been validated with identifying the clinical features that the model was picking up was the ones that have been known to be associated with path-CR in breast cancer. So, but overall, when we look at these, we found that the features together have a very good prediction for path-CR. Then we started looking at the pathology data. We used the pre-trained transformer model, and then we used multiple instant learning and deep learning to analyze data and with the dichotomized path-CR and no path-CR outcome. Here you see the heat map, attention heat maps showing where the model is focusing on. And we also did the pattern extractions, and this resulted in, and these are some preliminary findings showing us that in some subset of breast cancers, the model actually performs well and can predict the outcomes using only pathology data. This is the radiology analysis. We looked at the pre-treatment MRI data. We trained the segmentation, automated segmentation data, and also normalization of the images. Multiple different instances of the MRI were used, sequence of MRI were used, and then we used the deep learning model to predict the outcome in this group of patients. Very interestingly, these are, again, preliminary, but it seems like the model is performing very, very well in triple-negative breast cancer patients, but not as much in HER2-positive breast cancers, seeming that there might be some real data in the imaging predicting this type of outcomes, something that we're looking at much more closely now. Then we looked at genomic data, the patterns for this particular analysis. We looked at the variables and the genes that are already known to be associated with outcomes, and the signatures that are known to be associated with outcomes. And this is the data more or less predicting and confirming what we have known about genomics of breast cancer. The next phase of the analysis, we are doing multimodal integration. After we finalize the unimodal analysis, we are going to do the data integration, and we're looking at different models with early fusion, late fusion, or intermediate fusion to see which one has the best performance in this type of analysis. So with that, I would like to conclude, and just a couple of take-home messages. This type of integrated clinical genomic real-world data analyses are a significant discovery toolkit, and with the fact that we are using the clinical and genomic data more routinely in clinic provides an opportunity in this setting. There are multiple efforts ongoing, such as AACR Genie, looking at real-world data collaborations and harmonization of the data. This encompasses some of the biases associated with single institution, provide standard analytical pipelines to streamline this type of analyses, so on and so forth. And also, it's completely inevitable to use machine learning in the near future in this setting. So with that, I would like to thank all the collaborators and everyone who's done the work, and I'm not sure if we have time for questions.
Yeah. All right. All right, I don't see anyone at the microphone, but if you have any questions, oh, sorry, we have one question.
Hi, Josh Gruber from UT Southwestern. Really just curious about your data, the genetic interaction between BRCA2 and RB. Just curious if you can speculate a bit about why that occurs. I mean, is it a cell cycle checkpoint that needs to be bypassed in BRCA2 tumor genesis, or perhaps a difference in histologies, given that ER positive is much more likely with BRCA2?
Yeah, that's a great question. We think part of it is the fact that, in the BRCA2 mutant tumors, they already have loss of heterozygosity, so it's much easier for the tumor to lose the second copy of RB, and that makes it easier for the tumor, especially when you put a lot of pressure on with CDK4/6 inhibitors, and this is such a big resistance phenotype. Also, some of the analysis that I didn't show, HRD itself, it seems to be also inducing RB1 loss, and a lot of the mutations that we found in this group of germline BRCA2 patients actually had signatures associated with HRD. So it's both of them. We are looking at actually now more HRD genes to see if it actually, this phenomenon stands with HRD as a larger picture or not.
Got it, thanks.
All right, thank you. Thank you, Dr. Razavi. So our final speaker is Dr. John Irwin from the UCSF, who's going to be discussing advances in drug screening, both in the wet lab and virtually.
Thank you. Thank you for the invitation from the organizers to speak. Let's get this going. These are my disclosures. They're on the uploaded version. And these are my acknowledgments. This work is done at UCSF, and it's funded by the NIH and DARPA. So small molecule ligand discovery is a key important step in the long path of drug discovery. And what we're going to talk about today is molecular docking as a discovery technique. And what's new in our field in the last few years is the growth of the libraries of compounds that can be screened, shown by this chart here. And so we're going to take this tool, and I'm going to show you what you can expect from a docking campaign, what you can hope to achieve. And we'll do a deep dive, and then we'll ask, well, is bigger really better? Is it really worth all the effort that goes into screening these large libraries? Spoiler alert, yes. Okay, let me just introduce docking to you very quickly. Some of you, I'm sure, have seen this before, but you need a structure of a receptor. We live in the world of AlphaFold 2. I'm happy to say that AlphaFold 2 is often a terrific structure for discovery. There's a paper coming out from our group. The database is one of our main focuses of what compounds you screen. You only find the compounds that are in your library. The docking program takes the small molecules and it fits them into the binding site in many different orientations and conformations, sampling them and retaining the best one of each. Order of magnitude, about 1 million individual energy evaluations. And we then, using a physical chemistry scoring function, which necessarily has huge approximations in order for it to be fast. And from among the top high-scoring molecules, we pick the best scoring ones, we get them made, and we test them. And I'm going to tell you some of those stories. And what's new in our field is that as recently as 7 years ago, people were screening 3 million, and they thought that was a lot. And now we're screening this kind of number, and it's only going to go up. Where do you get billions and billions of molecules? Well, so it turns out that these guys in Kyiv, here's our interlocutor there, figured out a long time ago that if you make very small building blocks, we call these building blocks with synthetic handles, and you develop bulletproof chemistry, which is widely known, and then you can make, on demand, billions and billions of molecules by simply putting these together when the customer orders. And they figured out a way to do this with 90% success. And so we're all living in Enamines world right now. So here's 3 stories that show you, what you can hope for from a docking campaign. And I should stress at the beginning that there are absolutely targets where we strike out, and I'm showing you some good ones. So this is alpha-2A adrenergic receptor, the work of Alyssa Fink. This is the serotonin 2A receptor. Again, these are both GPCRs. And then the serotonin transporter, it's a transporter. Okay? And in each case, the structure became available to us. And we launched a campaign screening, tens of millions in those days, tens of millions of compounds. And from among the top 10, scoring molecules, we picked, 50 or 60 for testing. And from among those, we found compounds that were sometimes in the nanomolar range, sometimes only in the micromolar range, but not unusually in the nanomolar range, straight out of docking. And often, and in these three cases I'm showing you, we were able to get subsequent structural determination of the ligand that we predicted in the binding site, and we could superpose our prediction with the experimentally observed pose, telling us, cluing us in that we got the right answer for the right reasons. And you can see that sometimes there's some variability, but we'll often get the sort of the business end of the molecule, or we call it the warhead end. We'll get that right, but there'll be some variability. And these compounds can often, sometimes they don't even need optimization if they're really high affinity. Sometimes they'll need some analog by catalog to sample some structure-activity relationships to get a compound that's ready to go into animals. But in each of these cases, this work is all published, as you can see. The compound went into the animals and it exhibited non-opioid analgesia, non-hallucinogenic antidepressants, and in this case, a compound for depression and withdrawal. So this is a story basically where we're taking docking and we're going from a structure with a library of compounds. We're getting the compounds made. These are new to the planet compounds. We're checking to see that we got the right answer for the right reasons. Although, if we didn't, we might still go on, but we like to see that we got the right answer for the right reasons. And then we're putting it into the animal. Okay, so this sort of gives you a thumbnail sketch of what you can hope for from a docking campaign. I'd like to take a deep dive and look at one particular target. This is the cannabinoid 1 receptor, for which the structure is available. And cannabinoid 1 is, CB1 is ubiquitous in pain circuits. We absolutely need new pain meds. And it signals presynaptically through inhibitory G proteins. There's a large number of compounds that are all already known for the cannabinoid receptor. You might say, why are you bothering? There's endogenous compounds, there's compounds from plants, there's synthetic compounds, drugs, but what almost pretty well all of these compounds have in common is that they have this tetrad of symptoms. So in addition to analgesia, they also have catalepsy, they also have a temperature modulation and this locomotion activity. And so can I go back? Yes. So it's not a perfect correlation, but basically these four phenotypes travel together. And what we'd like to have is analgesia without these other effects. Okay. So this is the work of Tia in the lab. So in those days, we had 74 million compounds that were eligible for her screen, and she screened them against the library. And the first time she did it, she got 0. So I told you sometimes we strike out. We struck out completely on this run. What happened? Well, so here's a plot. This is a plot of, it's very, very small, I apologize, but what you're looking at, let me talk you through it. This is a molecular weight, and this is log P, so octanol water partition coefficient. And the purple shows you the history of everything, all the ligands that are known for CB1. And originally, we purchased what we thought was the correct thing, lead-like molecules. Everyone tells us, you should screen lead-like molecules. So that is down here. So the compounds were just too polar. This is a very greasy binding site. And when we instead selected from this area with these cyan dots, a little bit greasier and a little bit bigger, we were able to find 46 compounds that we liked, that Tia liked, and we tested and we got an 18% hit rate. Hit rate is number of compounds active divided by number of compounds purchased, not 74 million. And so these are some examples of the structures that Tia discovered in this work. And this one, the best of these compounds, we were able to optimize through a series of purchasing steps. So we could purchase from catalogs, from these enormous billion-scale catalogs, and through, you see, by turning the fluorine into a trifluoromethyl, and then a little bit of a rearrangement over here, she got down to a 1 nanomolar compound. And these have, very good kinetics and behavior. They're all quite kosher. And so that compound, which we were able to subsequently determine the crystal structure, cryo-EM structure. And so this shows you the electron density, and this is a superposition of our pose. Again, it's not a perfect, angstrom for angstrom superposition, but it gets the important features of the molecule right, and we think we're getting the right answer for the right reason. Okay, so it's all very well to have a compound that you've predicted and that binds at 1 nanomolar, but is it any good? So the first question is does it even get into the mouse? And does it get into the brain? So this is the work in the lab of Alan Basbaum at UCSF. And so these are the charts this is over time, and the compound does get into both the plasma and the brain, and it seems to last about 3 or 4 hours, and it's distributed, it's biased towards brain. So that's good, cerebral spinal fluid. So that's a good thing for pain. And then does it actually work? And so remember, this is a compound that we've just found from docking, and we're just putting it into a mouse. And so what you can see that there's this is our compound, and we're comparing it to a Pfizer control that's widely used in the field, this one here. And so what you can see from our compound is that there's dose response in the tail flick assay. There's several different versions of this, but a lot of detail in the paper. But it's providing analgesia. This is the complete Freud's adjuvant. It's an irritation model. You inject it into the mouse, and then you put this metal into the mouse's paw, and you see how it withdraws. So this is the withdrawal latency, and you can see that our compound is providing analgesia in this assay, and this is an acetone test. So these are various nociceptive behaviors that the mouse does, licking the paws, lifting the paws, and so on. And so in each of these cases, so the compound is active with a lot of different phenotypic effects for pain. And then remember one of the problems I told you about in the tetrad was catalepsy. So this is the assay for catalepsy. So I'm going to show you a video in a second. And so this chart summarizes it. So over time, 30 minutes and an hour, and this is the latency to movement. So we're going to give the mouse Haldol, which is not a CB1 ligand, but it induces catalepsy. So let's see, we put the mouse on there, and this is a cataleptic mouse. Doesn't move. Okay, then we're going to give it the Pfizer compound, and you see the mouse moves a little bit, but it doesn't move very much. And then we're going to give our compound at an analgesic dose, and the mouse is totally normal, and it climbs off the wall. So this is, again, the work of Alan Basbaum. Rotarod, this is the sort of the Canadian roller of logs. So this is a measure of sedation, and so this in a dose-response fashion, and eventually the mouse starts getting sedated and it falls off the log, but it doesn't until way past the analgesic dose. Whereas with the Pfizer compound, it does. It starts falling off earlier. And then, so now finally, if we're going to compare our compound to the Pfizer compound, which is not necessarily the standard in the field, but it's a widely used reference. And if you normalize all of the effects, sedation, catalepsy, and so on, to 1, our compound has significant analgesia and definitely reduced effects along other axes. So this is an example of new chemistry provides for new biology. By finding completely new chemotypes, we've gone through what we believe are the same targets, CB1 and we are seeing new biology, new effects. Okay. So I remember I told you I promised I would come back to the question of, is bigger really better? And I've got a story to tell you very quickly. This is AMP-C beta-lactamase. It's an antibiotic resistance target. We actually did a screen against AMP-C back in 2018, and at that time the library only had 99 million molecules, so 0.1 billion. And so at that time we tested 44 compounds, which admittedly is not a very big number. And we measured hit rate. And now our library is 2.1 billion, and we were able to test 1,500 compounds. We can still get a hit rate. And so here's how they compare. So in the first screen, there were no hits better than 1 micromolar, and a couple when we do the 2 billion. Better than 10 micromolar, the hit rate is almost 9% when you're screening 2 billion. It was about 2% when you're screening 0.1 billion. If you count up to 100 micromolar, and we believe these compounds are all legitimate, they've got good dose response curves, they aren't really high affinity, but they could be optimized. The hit rate goes up to 30%, whereas in the first screen, much, much less. And so from this trend, what we're seeing is that when you screen a bigger library, you've got more to choose from. And so now you can find new chemotypes that you can give to your medicinal chemists for optimization towards candidacy, ideally. Okay. And the last data slide I'll show you is with the hit rate as a function of the DOC score. This is our DOC scoring function. And whether you look at 200 micromolar, 100, 30, or 10, the hit rate falls off as the score gets worse, meaning that DOC can somehow rank the library from best to worst, at least at some level. The resource that I've been screening, it's currently at around 6 billion molecules. It's free. You just come to our website and you can access it. It's also available in AWS. And so the thought I'd like to leave you with is that if you've thought about docking before, or you've been, it didn't work for you, maybe give it another look, because chemical space has really grown lately. It's only set to keep growing, and there's tremendous opportunities for discovering new chemistry for new biology. Thank you.
Thank you so much. Thank you so much, Dr. Irwin. I think in the interest of time, we'll have questions outside the hall and let the next group come in. But please join me in applauding all of our excellent speakers. Thank you.